All right. Yeah, today's lecture is on multivariate pattern analysis. And the focus of this multivariate pattern analysis lecture will be mostly on fMRI as a functional MRI. This is a little bit out of like the normal schedule because we had to shift things around a little bit. So some kind of prerequisites you might not have really like learned yet or you haven't heard about. But I try to make it like as accessible as possible. So just to start off with a very general introductory example is most of you were sleeping just like half a day ago, and many of you were probably dreaming of something. You might not be aware of it, but most people dream all the time when they're sleeping. And then you wake up and sometimes you recall what you were dreaming of. But like dreaming is a very personal experience. It's something that you wouldn't necessarily want to share with everyone else. And it's something where you think, okay, that's something, it's personal and no one else can access it from outside. But now just imagine that someday in the future, brain scientists can actually read your brain activity while you are sleeping, and then they can kind of reconstruct the content of your dreams. So now this might sound a bit like science fiction, but in actual fact, there was a study that was examining exactly that kind of question in 2013. So what they did is they had put people in an MRI scanner, and they had them sleep. It was monitored with EEG, so they really knew that they were sleeping. And then they woke them up, and they woke them up a lot of times. So every time they woke them up, they asked them, what were you just dreaming of? And then the participant reported what they were just dreaming of. So now what you could do is you can take the brain patterns that were recorded while the participant was still sleeping, and you can see if you can actually decode what the content of the dream was. And it turns out with a small number of categories that the authors were using, they could decode from like higher visual cortex with an accuracy of up to like 75% what the participant was dreaming of. So I like using this example for multiple reasons. First of all, it tells you something about like the power that you can have when you're using multivariate pattern analysis for looking at brain representations and for studying like how the brain is working. But it also tells you something about potential ethical implications, right? So if we use these methods for, let's say, lie detection or other ethically more challenging topics, like what are the problems that come with this use? But this talk is going to be focusing mostly on the method itself, and I will not be talking so much about the ethical implications, but I'm more than happy to discuss about those afterwards. So just to start off more generally, like what is multivariate pattern analysis? And it's kind of difficult to define, so people have kind of avoided defining what multivariate pattern analysis is. So I just try to wrap it up myself. And for me, it is like the combined use of multiple variables, and that's the most crucial part here, the multiple variables part, that are measuring the brain. So for example, you could be taking the bold signal from multiple voxels to predict, which would be decoding, or to characterize, which would be other methods that we'll be talking about later, to predict or to characterize states of the brain. So the combined use of multiple variables is what makes it multivariate. So why would you want to use multivariate pattern analysis? Well, it turns out that another thing that's the most crucial point is that it often gives you higher sensitivity as compared to classical univariate analysis. So in other words, it makes it easier for you to find significant results, and that's obviously what many people in science are interested in, is having significant results that they can later publish. So just to give you an example, looking at the representation of perceptual choices, there's a study that was conducted in 2009 where they looked at two different choices that participants were doing, and here you can see where you find discriminatory activation if you're using a classical univariate approach between the two different choices. Now, if you're using a multivariate approach instead, what the authors were finding is much more widespread discriminatory patterns of activity. And if you look at the p-values here, this was like a p-value of 0.01 uncorrected, so there was not really much going on here anyway, whereas this one over here has is 2 orders of magnitude smaller. So this really emphasizes the power of using multivariate pattern analysis. But on top of that, so on top of only like getting the higher sensitivity, you can also get a higher specificity. So which allows you to study representational content rather than general activation. So what do I mean by the higher specificity and the possibility to study the representational content? So to just take the example of the representation of orientations in visual cortex. So we all know that visual cortex is responding to visual activation, to visual stimulation. So when you stimulate an animal or a human visually, then the visual cortex activity will increase. And if you increase the contrast, the activity will go high as well. But that doesn't really necessarily tell you anything about what is represented in the primary visual cortex. So is primary visual cortex responding to changes in contrast? Yes, it is. But if you ran like a classical univariate analysis in primary visual cortex and you were interested in looking at differences between different orientations, would you still be able to find them? So we all know that primary visual cortex is representing different orientations, but is that something that's accessible with classical univariate analysis? Well, most people say it's very difficult to access, but with multivariate pattern analysis, what you can do is you can run a classification and you can decode like these different orientations, and you can see that even using a very small number of voxels, in this case like up to 50, you can already decode what orientation a person was shown with an accuracy of around 75%. So in other words, what MEPA allows us to do is to go like one level lower. So let's take the example of object selective cortex. Rather than finding whether a brain region responds more strongly to objects than non-objects, you can now tell whether this brain region actually carries information that discriminates between different types of objects. So you can go to like one level lower from just like talking more generally about objects to like specific types of different objects. Okay, so now how does MVPA actually work? And I think there's in general like 3 different principles that are underlying MVPA, and that is not only for decoding but also for representation similarity analysis. And that kind of breaks it down to make it really easy to understand, and it actually is really not that difficult. So the first principle is that weak information can be combined across voxels. That seems to be quite obvious. So in other words, multivariate analysis can enhance the signal. So let's say you have one voxel that discriminates between so you're looking in one voxel to discriminate between condition A and condition B. You look at the activity and the activity difference is not significant. Now you look at another voxel and again you measure condition A and condition B, and again there's no significant difference. But now what if you were to optimally combine those 2 voxels together? So let's say you have voxel 1 minus voxel 2. In this case, this difference would add up, and you could see that, well, in this specific example, the difference could now become significant. So in other words, what MEPA helps you to do is to find this optimal combination of these different voxels in order to enhance the signal that is already present in the data. Okay? So another important principle is that the covariation of voxel information can be used. So in other words, multivariate analysis can suppress noise. And this noise suppression part might seem a little confusing at this moment, but I'll explain to you in a second why this is noise suppression. So just say, like, again, we have 2 conditions, A and B. And what I'm plotting here on the x-axis will be the activity, and it's going to be a histogram. So what we're plotting on the y-axis is the frequency of how often this measure that we're going to get in this voxel. So this is like a histogram that we get for the activity of condition A. And that's the histogram that we get for the activity in condition B. And you see they overlap very strongly. And in another voxel too, you might actually find something that looks very similar. So you find activity for condition B looking like this, and for condition A looking like that. Okay, so now what happens if you combine the information from those 2 voxels together? So you can plot voxel 1 along this axis, and voxel 2 activity you can plot along this axis. So again, like the differences for each voxel alone don't really tell us very much. But now if you look at the covariance between those 2, what might happen is that you actually find something that looks like this. So now in this case, you can actually see that even though each individual voxel does not allow you to discriminate very well between those two conditions, if you look at both voxels at the same time, you can perfectly discriminate between those two. And this has to do with the fact that voxel 1 and voxel 2 are negatively correlated, which you can see by this like negative correlation here. So this is also what I meant by multivariate analysis can suppress noise. But to give you just like a better example, a more realistic example, I'm going to take just like some simulated activity. So let's say activity looks like this. So I'm sorry that you can't see the colors so well because the projector is not working well, but we have two different conditions where in one case it's like active and the other one is not active. So this would be the true activity that we know is really going on in this brain. And now we're measuring from a voxel in the gray matter. And the measure, what we will end up getting is a noisy version of the true activity, obviously because there's noise in the brain, right, in the measurement. But on top of that, we'll also be measuring a white matter voxel. And now what the white matter voxel is it's going to be pure noise because there's no neurons in the white matter. So this can only be measuring like noise all by itself. Now, interestingly, since the noise is going to be correlated across different voxels, and that's something that we know, the classifier can make use of this. So now if we again plot voxel 1 against voxel 2, what you can see here is in voxel 2, we have no discriminatory information at all. Whereas in voxel 1, we do have this discrimination, but the discrimination is not perfect. Now what voxel 2 allows us to do is essentially like we can subtract the noise out of this voxel here, and by doing this in an optimal way, we can then like really achieve, in this specific case, perfect classification, right? So this is really something I think that needs to be emphasized more in general, is that classification essentially does denoising for you, okay? Good. And a third principle of how MVPA is working, and that's probably more related to representational similarity analysis, is that the information that's encoded only in a distributed activity pattern is now becoming accessible. So again, to give you an example, this time we have 3 different conditions, and what you see is like, okay, well, voxel 1, like for condition B, we have a stronger response than for the other 2 conditions. Not really sure what that means, but it seems like voxel 1, condition B is encoded more strongly, and in voxel 2, condition C seems to be encoded more strongly. But if you look now again at like the 2 voxels plotted against each other, what you see in reality is actually you have just 3 very different categorical responses, where in condition A, you guys have always a low response. In condition B, you have a high response in one voxel and a low response in the other voxel, and in condition C, you have a high response in the other voxel and a low response in the other. So in other words, if you were just only looking at one individual voxel, you would never be able to discriminate all 3 of these from each other. Whereas if you look at both voxels at the same time, you can now distinguish those responses to all of these different conditions. And that's also quite telling if you think about like information processing. So we know that a bit has like is either 1 or 0, but as soon as you have 2 bits, you have 4 possible combinations. If you have 3 bits, you have 8 possible combinations. So there's a combinatorial explosion for the possible states that you can have when you're measuring more than one piece of information at a time. So what we have here is each voxel you can kind of treat like a bit, although we have more continuous dimensions here. So if we combine multiple voxels together, there's many more different possible states that can be represented in an activity pattern. So this is like the third like principle of how MVPA is working. Okay, so one important distinction that I like to make is the distinction between activity and information. So activity is like the classical way of thinking about like how the brain is working. We normally think that activity tells us something about how processes are represented in the brain. So if a brain region is more strongly active, it's more involved in a cognitive process. That's our classical thinking about brain activity. So just to use the example of working memory, while we think that the frontal cortex the prefrontal cortex seems to be involved in working memory just because it responds more strongly in a working memory condition as compared to, let's say, a baseline condition. But now what this doesn't tell us really is what is being represented in frontal cortex. So I'll just give you like two possible explanations. One is that frontal cortex is re-representing whatever you are holding in your mind at the moment. So when you're trying to hold something in your working memory, let's say a phone number, then the frontal cortex would be carrying another representation of that phone number. Alternatively, though, what the frontal cortex could be doing is it could just be helping you maintain access to a representation of that phone number elsewhere in the brain. So you just need the frontal cortex to suppress other activity elsewhere in the brain. It's just like one possible explanation. So this is something that the classical activation view cannot really address. But with multivariate decoding, you can really ask the question, well, what's like represented in that part of the brain? So in other words, like the information view tells us something about the representational content. So there's a study that we conducted in 2012 where we were showing people like different artificial stimuli. We're using artificial stimuli to make sure that people are not using a semantic representation here. They're not like verbalizing internally. And we wanted to know if indeed like frontal cortex or what part of the brain is representing the content of the memory. So while people were holding these patterns in mind, we were looking at the brain activity patterns, and what we found is replicating what other people found previously is that primary visual cortex was representing those patterns, but on top of that we also found that the posterior parietal cortex was carrying a representation that was discriminating between these different patterns. So interestingly, though, we did not find any information that was discriminating between those different patterns in the frontal cortex. So this would support the idea that frontal cortex is not carrying a re-representation of the perceptual input, but it's mainly like having a role in like maintaining information elsewhere in the brain. Okay. So just to give you like a bit of a hunch of what I mean by the difference between activity and information. I just want to give you like a thought experiment. So let's say we have here the activity that we're measuring, and in one case the participant is presented with an apple, and in another case the participant is presented with an orange. And now what you find is you find a stronger response to the apple as compared to the orange. According to the classical activation-based view, we would now say that this voxel, or this brain region, is responding selectively to apples. So it's an apple-selective brain region. Okay? So now what happens if I add more fruit in, and this is just for the sake of the example. So what might actually happen is now we add a pear and we see, oh, there's actually a stronger activity also as compared to the orange, and we add more and more fruit. And now actually what we find is that in general, the activity is always higher for all types of fruit as compared to the orange, okay? So according to the classical approach, what this would mean is that the brain region responds to all fruit but to oranges, which sounds like a little specific or a little awkward. However, what the information-based view in MVPA would be is, well, this brain region is just carrying information about oranges. The way it's coding oranges is by deactivation. And it's coding information of oranges also always relative to other fruit. So if there was other stimuli, if there were other stimuli that were presented here, maybe the response would be very different. But in general, what this idea is telling is, well, activity doesn't necessarily have to be, or information doesn't necessarily have to be represented by higher activity. It can actually also be represented by lower activity. Okay, so what are like the levels at which you normally carry out an MBPA analysis? And in general, I would say there's like 3 different levels, at least for fMRI. One is what you can do is you can just look at the whole brain at the same time. So looking at the whole brain at the same time will give you access to all the information that you have present in those patterns of brain activity at the same time. So you make maximal use of all the information that you have in theory. Well, what in practice that gives you is just it gives you one number. And this number will tell you, well, that's the accuracy, that's the decoding accuracy, that's how well I can extract information from this brain, but it doesn't really tell you where the information is coming from. There's people who try to figure out ways in which you can make this interpretable, but it's very difficult actually to do so. And let me see. And another issue is that now you have so many different voxels. So there's like such a high dimensional space that we're dealing with that actually turns out to be quite difficult to find a really good solution. Because like, just think about it, you only have a handful of activation patterns, but you have maybe like 100,000 different voxels. So you're now dealing with 100,000 dimensional space, and you have only a handful of like points in this space. How are you ever going to be able to like describe the space appropriately? Well, it turns out you can't, and that's also one of like the potential downsides of looking at all of the voxels at the same time. So what you want to do is you want to find ways to reduce the complexity of the problem. And one typical approach is to take just a region of interest. So you say, okay, I'm specifically interested in looking at this part of the brain. Now we only have like 500 voxels, so this drastically reduces the complexity. And another good thing is that it's much easier now to localize where the information is coming from. So now we have one value per region of interest. You can also take this to the other extreme, and now you can do something that's called a searchlight analysis. So in a searchlight analysis, you take a region of interest that's spherical, and you will calculate locally multivariate pattern, conduct a multivariate pattern analysis locally. You get one result back. You store that in a separate empty brain. You repeat the analysis for the neighboring voxel. You store that as well in that empty brain, which now contains two numbers. And you repeat that analysis for all possible spheres in the brain. So you move that little like spotlight or searchlight around. And now what you end up getting is you get a map. You get a map of values, which for like multivariate decoding would give you like something like decoding accuracies, and for representation similarity analysis could give you like a correlation coefficient to whatever you're interested in. So the nice thing about this approach is it gives you much more fine-scale picture of where the information is represented. The downside is you're not using all of the information that's available, and also you have like more of a multiple comparisons problem here because you again like they have 100,000 tests that you need to run later. Okay, so what are the main approaches that are being used in multivariate pattern analysis? I guess most of you are aware of these, but just to point them out. On the one hand, there's multivariate decoding. So that's what I was also using in an introductory example, where you have these different activity patterns. So you have voxel 1 activity and voxel 2 activity that you could plot against each other. And what you do is you try to draw a line between those two. This would be like a classifier. A classifier is just a function that allows you to separate those different conditions. And now what you can do is you can see if you can predict other data that has been left out. So what would be the prediction for this little triangle here? Well, obviously it would be red. We don't know if it's correct, but this would be the prediction. And for this little triangle here, the prediction would be blue. The other main MBPA approach is representational similarity analysis, and I will go into more detail about what representational similarity analysis is a little later. So, but first, coming to multivariate decoding. So, multivariate decoding has two main goals. There's two different ways in which you can apply multivariate decoding. One is for prediction and one is for interpretation. And just to explain to you what the difference between those two means, just look at the top here first. So let's say the goal is prediction, and we want to be able now to predict from patterns of brain activity whether this pattern is distinguishing between cars and chairs. So we are going to run multivariate decoding, let's say in this little region of interest. We can now decode, so we find information that distinguishes between cars and chairs. So we can conclude, yes, we can decode cars versus chairs. When the goal is prediction, that's all you are after. Now, what is the new thing and what made like multivariate decoding so popular is the interpretation view. So we doing the same thing. We're asking the same question. Can we like decode multivariate from the multivariate patterns in this brain region, whether there's a car or a chair? And once we can find this difference, we don't stop just saying yes, it can be decoded. We actually go one step further. What we now say is, well, since we find discriminatory information in this brain region about cars versus chairs, well, that obviously has to mean that this brain region is representing objects, okay? Because if it wasn't representing objects, it would not be able to distinguish between those two conditions. And if you do that for many more different objects, that is actually potentially like a valid conclusion that you could draw, although obviously you have to take other alternative explanations into account. So for prediction, the goal really is to maximize future correct predictions, and you can really use any type of information as long as it helps you increase the accuracy. So one example that here for NIH might be kind of important is medical diagnosis. So people are working very, very hard on building machine learning applications that allow you to decode from medical images if there's something wrong with that person's brain. And there's actually quite a lot of progress being made right now with the advent of deep neural networks that can often compete quite well with at least beginners and in medical training. So medical diagnosis is one possible application of decoding. Then there's brain-computer interfaces. Well, if you can control, or if you can steer like a wheelchair just by using your brain activity, well, that's going to be a very useful application, obviously. But now the maximization of the correct predictions becomes also very important, because if there's just one mistake that you make at an intersection here, that can have obviously lethal consequences. And that's something that, that's an example that obviously matters right now when you were thinking about self-driving cars. Another example is lie detection. So you could imagine that one day we are able to really tell whether that person is telling the truth or is lying, and we could use that as evidence in front of a court. If it turns out that our methods are really very reliable at telling the difference between the truth and a lie, is that something that we want? Is that some prospect where we say, okay, we're going to put people we put people in jail who really belong there, and we can tell with a very high probability that they were lying or telling the truth? Or is that something that has more of a problem? But these are possible applications. Another one I can think of is neuromarketing, but there doesn't seem to be that much progress in this area yet. So this is like the first goal, prediction, but like what most of us doing like more basic brain research are interested in is using decoding for interpretation. And as I was saying before, the goal for decoding for prediction was to maximize the information that is available. Whereas here, for the goal of interpretation, all we need to say is there information about X, Y, and Z? So we don't actually have to find like very, very high decoding accuracies. It's actually sufficient to show that we find above chance accuracy, just statistically above chance. So just to give you an example here, so this is a study that was conducted, I think in 2005, where people were showing like different orientations, and they were presented them either consciously or unconsciously using backward masking. And then the authors wanted to figure out if you can actually decode from the patterns of brain activity, even for the invisible orientations, if you could still decode those from primary visual cortex. And for the visible condition, you find an accuracy of about 80%. Whereas for the invisible condition, the authors found an accuracy of a little lower than 60%. So now you could say, oh, that's a really bad classifier. It can only decode like 60% correct. But what this actually tells us is, no, actually this is great, because even though the classifier itself is not doing a fantastic job, it tells us that, well, the brain definitely is carrying some information about those orientations. We're just not very good at extracting it, but it doesn't matter as long as it's above chance. We're better than guessing. So if there is information about them, maybe one day we can build a classifier that's better at extracting that information. But as long as we find that information, that's actually great. So now we can say, yes, the brain is representing orientations even when they are invisible, at least in primary visual cortex. And here's just another study where you can even see where people are taking this a bit more to the extreme, where now you can they are interpreting interactions that are happening even like for an accuracy of 52%, where it all depends really on the statistical power that you have for testing these differences. One thing I haven't mentioned yet is that, well, not all information sources are okay. Like for prediction, you can really use pretty much any information that you want, but here we really have to make sure that there's no confounds. So obviously, you want to make sure that that it's not like some basic, I don't know, muscle movements or something, or eye movements that are explaining your results. Okay, so just to give you like an idea of how multivariate decoding works, how does multivariate classification work? I just want to give you like a very simple example. And I used to study perceptual decision making, so I like to use the example of making choices. Okay, so let's say we have this part of the brain that we're interested in. And we are interested in knowing whether people are making we're trying to decode information that will tell us about whether a person is making a choice to the left or a choice to the right. So what we're going to do is we're going to take the patterns of activity that we extract from those parts of the brain, and the color now is coding the level of activity. So that's the way that you would normally see voxels in a black and white image, right? So that's the first step, is you extract the patterns of brain activity. Now, when you extract them, your software will automatically vectorize them. So what you want essentially is, per condition, you want to have a pattern vector. And what's really important here is that the order has to be maintained. Obviously, like when you extract the pattern of activity here, this voxel here has to correspond to this one over here. So in other words, if this voxel is this one here, then this one has to be this one here. Okay? So for the vectorization, you really have to make sure that it's always done exactly the same way. Now, each one of these patterns is maybe, let's say, has like a length of 100. So what we're dealing with is a 100 times 1 vector, which means that we have 100-dimensional, we're dealing with a 100-dimensional space here. And it's not very easy for me to plot 100 dimensions on a PowerPoint slide. So what most of the examples normally do is they only pick like 2 dimensions. So what I'm going to do is I'm going to illustrate what we're doing in the decoding only in 2 dimensions. So let me focus in on those 2 here. So what we have now is 2 activation patterns. 2-dimensional activation patterns. And again, what I'm going to do is I'm going to plot voxel 1 against voxel 2. So this is voxel 1, this is voxel 2. So now for the blue condition, what we expect is a low activity in voxel 1 and a kind of intermediate activity in voxel 2. So we get a dot that's sitting exactly here. For the second condition, we expect a high activity in voxel 1 and a low activity in voxel 2. So we expect a dot that will end up over here. So in other words, what this dot is this dot is representing a pattern. Another way of thinking about this is that a pattern is a dot in high-dimensional space, okay, or just a vector, essentially. So now if we don't only have one measurement of each one of those, otherwise it would be kind of difficult to look at, to build like a classifier. I mean, we could, but it wouldn't make a lot of sense. So we're going to have lots of repeats of the different conditions. And now once we have a lot of repeats, we can see some sort of pattern emerge. And now what we do in the next step is what's known as training a classifier. So we'll try to find a function that allows us to optimally separate between those 2 different conditions. And then once we've done that, we can then go ahead and we can assess the utility of this classifier. Now, most of you have heard that we talk about training and testing a classifier. So I just want to engage you as well, and just want to figure out together, why do we actually train and test classifiers? So I just want to give you this example here. And I would like to ask you, do you think it's possible to build like a classifier that can perfectly separate between the red and the blue condition? So hands, raise your hands if you think yes. Raise your hands if you think no. Well, I'm going to tell you that it is possible, and this is the classifier. So I've just built a classifier. It's a highly nonlinear classifier, but you can see I can perfectly distinguish between these 2 different conditions. So now, what is wrong with this? Like, what's wrong with building a classifier like this? Can anyone give me an answer? Okay. So it doesn't generalize. What does it mean it doesn't generalize?
If you were to have another data set, you would be able to predict on that.
Great. Okay. So let's now imagine that we had another data set. Okay? And this is the classifier that we built. And now we want to make a prediction. I'm not going to reveal to you what the true labels are in here. I'm just going to apply the same classifier, because if it's a classifier that is a general purpose thing, it should also apply to new data. And I'm going to apply it to this part. And what you can see now is that it doesn't do well at all, right? And the reason it doesn't do well at all is, well, this is pure noise. Like, there wasn't any structure in the data to start off with. Like, I mean, I was able to fit something, but you can see that it doesn't really generalize to any other data. So what this is supposed to tell you is, well, what we're trying to find when we're building a classifier is something that allows us to generalize to new unseen data. We want to find a function that is complex enough to describe the data that we have available, but not so complex as to fit to the noise that we have in the data. So if I give you pure noise, your classifier will end up giving you a kind of random response. And then you also, when you test your classifier on left out data, well, the result will not look very good. So the goal of classification is to find a model that generalizes beyond the noise in the data. And that's a really important take-home message, the generalization. And the way of testing the generalization is to test the classifier on new data, which is also known as out-of-sample generalization. Okay, so now, but there's a problem now with that, because we've measured our data, and we want to use as much data as we can for training a classifier, obviously, because we want to make maximal use of the data that we have available. But now if I split my data, I mean, I'm always going to lose part of it if I have to use part of it for my testing, right? So what's a good way of like trading off between training size and testing size? So people thought a lot about this, and one way that people came up with that is actually quite a clever way is to reuse data for training and testing. And that's what's known as cross-validation. So please raise your hands if you know what cross-validation is. Okay, that's good. So half of the people already know it. So in cross-validation, you take all the available data that you have, and what you do is you separate the data into multiple parts. And this separation process is also, these different parts are now called folds. So what you have is you have training folds and validation folds, or testing folds. So let's say we split it into 5 different parts. Now we take, essentially we have 80% of our data available for training, and 20% of the data are available for testing. Now we run our classification, we make our predictions, and now in this case, 77% of the predictions were correct. So that's great. And now what we do in the next iteration is we're going to take another one of those folds and leave that one out, and we're going to repeat the classification process. So we're going to be reusing these 60% of the data in both cases, but we still have the independence of the out-of-sample prediction by always using a separate test set. So in this case, let's say we have a prediction of 83%, in this case maybe 71, in this case maybe 79, and once we've repeated the process for all of these different iterations, you see that due to random noise we always get like slightly different results. What people then normally do is just average those results together. So that would tell you something about the general predictability in the data set that you have available. That's the approach that's commonly used in fMRI. If you're actually a machine learning person, you normally have so much data available that you don't necessarily have to run cross-validation externally. So often machine learning people just have like a separate test set, and then they maybe run cross-validation internally to optimize some parameters. But we often in neuroimaging don't have so much data, so we just use cross-validation in general also for testing the model itself. Okay, so just to give you an idea of like typical linear classifiers that have been used, one is a very simple classifier. It's called Gaussian naive Bayes classifier. I mean, the term naive is also telling you something about the simplicity of it. Essentially what it's doing is it's looking at each voxel individually. It's calculating, based on the training data, a probability density function of a value occurring there. So the probability for this voxel here of red here is a lot higher than the probability for blue, obviously. Whereas here, the probability of blue is a lot higher than for red. So when you have a new data point that ends up right here, what you're going to do is you're going to just take the product of the different probabilities. So you're going to say, okay, so the probability of it being red is like this high here, and but it's quite low here. So we take the product of that, so this is the probability of red relative to the probability of blue. But you can see here the probability of blue is pretty much 0, and here is also like almost 0. So we can be extremely certain that this will be a red point. Okay. So since we are looking at each of the voxels individually, we're ignoring the covariance between those different voxels. Another approach is known as linear discriminant analysis, which is actually quite related to the Gaussian naive Bayes approach, but it does take into account the covariance between the different voxels. So what we're calculating is just like the general correlation between those two voxels, similar to the denoising approach that I was talking about earlier. And so we're trying to find like the for linear discriminant analysis, trying to find like the centroid of these like multivariate Gaussian distributions here that are supposed to have like the similar covariance structure. And then we build like a classifier that lies just right between those two, and carry out the classification in that way. And probably the most popular approach in multivariate decoding is using a support vector machine. Support vector machine is actually quite simple. What it does is it's first of all finding like a line or a plane, or in more than like 3D a hyperplane that distinguishes between these different conditions, so that separates them. And then what is being done is it tries to maximize a margin between those two. So, in other words, try to make like a border that's as thick as possible that you can fit still between those two different populations. So what this does is it will end up like always having some of those data points as edges, and they are supporting the solution to our classification. So that's also why they're called support vectors. So these are the support vectors, and that's why it's called a support vector machine, because it tries to find these vectors that are like at the margin, and then you carry out the classification based on the hyperplane that you find here. What is actually really cool about all linear classifiers, and that's also like something as a bit of a take-home message, is that the formula is actually always the same. So the formula is always that you have a weight that you multiply with the data points. I can't go into details about why this is the case, but you're always trying to make a prediction. Which is essentially the formula is always the same. The only thing that's different between the different classifiers is how you arrive at those little W's here, the weights that are telling you what's like going to be the orientation of this line. But in general, the formula is always the same. Okay, I'm going to skip over the correlation-based classifiers just for the sake of time, and go directly into representational similarity analysis. So representational similarity analysis is a bit of a different approach, but since this is like an MBPA lecture in general, I decided to include it as well. So representational similarity analysis, I'm just going to give you the gist of it because there's a lot to this as well. What you normally do is you have a stimulus that you might be interested in, and again, what we have is you have an activation pattern in response to the stimulus. So we have, like, let's say, an activation pattern in response to a hand, or in response to, let's say, an umbrella, and the activation patterns look very different. Now, what you do instead of looking at many of these different patterns and calculating a classification hyperplane, is what you actually do is you compute the similarity between them. So for example, you could correlate those two patterns and you can see how strongly they're correlated. And if they are very strongly correlated, they'll be very similar. As when they have a very low correlation, they will be very dissimilar. And we're interested in the dissimilarity between them. So then what you do is you do that for all your different conditions. So in this specific matrix here, we can see, well, an umbrella and a hand have quite a high dissimilarity, whereas in this specific example, I don't know why, but the umbrella and the pair have a very low dissimilarity for whatever reason. Or the hand and the, is that an ostrich? Yeah, I don't really know. So this doesn't make a lot of sense, but that's just what this example is showing. So what you're doing is you're calculating the pairwise correlation between all of those different pairs here, and you end up getting such a matrix. And then what you can also do is you can calculate like a similar matrix for behavior. We know for behavior we can ask people how similar are objects to each other. And you can ask humans, well, how similar is like a face to an ostrich. You can get a similar type of dissimilarity matrix. And now you can relate those dissimilarity matrices to each other. You can see in this example, there is some structure that they share. So maybe this tells us that this brain representation is somewhat similar to the behavioral representation. But you can also do other things. You can get like some stimulus description by looking at the pixel values, for example, and calculate like the pairwise dissimilarity in terms of just like the pixel values. Or you can have some sort of computational model that looks at the representation of these different objects, and you can relate them all to each other. So that's essentially the key advantage of using representation similarity analysis, is that you can relate these different methods of measurements like to each other. So this is just to give you like a general idea of what representation similarity analysis is, but just to also give you a bit more of an idea of why you actually do this. Like, why would you calculate these similarity matrices? We just go one step back. So when you do different measurements, what you could do is you have like an electrode in a monkey recording. So let's say you have an electrode X and an electrode Y. And in a human fMRI study, you might have a voxel A and a voxel B. And you just plot them against each other, and you're showing them human different stimuli. And this is what you find. So you find that voxel A responds a little bit to this car, and voxel B also. And you find different responses for, let's say, a table and a chair. Now, you can also look at the monkey recordings, and let's say this is what you find here. So you find a very different response for the car. So now this electrode Y responds a lot to this car, and electrode X doesn't respond so much. And that's obviously the case because the electrode X doesn't have any correspondence to voxel A at all. They're completely different measurement spaces. And it's also measuring at a completely different resolution. But one interesting pattern can emerge, and that's also something that some people have reported, is, well, when you look at the patterns that you see in here, what you find is that, well, but the table and the chair seem to be closer to each other than to the car. And maybe in the monkey recordings, you actually find the same. So there's no way we can directly relate those different measurements to each other. We have the electrodes that are measured like the millisecond scale, and we have the voxels that are much, much larger spatial scale and are measured like at the second level. But we can still see that, well, there's a lot of similarity in between those two, and some dissimilarity to those activity patterns here. So what's a way in which we can make those, we can relate those to each other? And that's where like representation similarity analysis would come in. So now you can take, for example, the representation distance, so for example, the correlation, so 1 minus the correlation, or you can take the Euclidean distance, and you create this representation dissimilarity matrix. So now you see, well, the dissimilarity between a chair and a table is lower than the dissimilarity to a car. And you can use this, and you can maybe calculate the same for the human fMRI. And now what you see is that these dissimilarity matrices are actually identical. So in other words, while at the measurement level, you find very, very different responses, you can see that the overall pattern of response is actually quite similar, and that can be captured quite nicely by the representational distance between those different patterns of activity. And then what you do is with representational similarity analysis, you're relating the patterns at the representational level to each other. So does this actually work? I mean, this sounds a little crazy to start off with, right? So why would you actually be able to measure something that looks similar in the monkey electrodes as compared to the human fMRI? But it turns out that in 2008, and that was the first representation similarity analysis study conducted by Kriegeskorte, who also strongly promoted this approach. This is for 92 different objects that are presented. And what you can see is actually for monkey IT and for human IT, you see the general structure of those matrices is remarkably similar. So in other words, measuring with electrodes from monkey IT and measuring with fMRI from human IT can still give you very, very similar responses at the representational level. So that really gives you some insight into the power of using representational similarity analysis as a measurement tool if you want to compare, let's say, between monkey and human data, or you can also create these matrices using computational models like deep neural networks, or you could look at human similarity judgments. This is something that I'm working on myself right now. So there's lots of different things you can do. So it's quite a powerful method. Okay, so just before we wrap everything up, Peter Banatini asked us all to add some recent challenges or recent issues that people are still dealing with the methods in here. Now, the difficulty is that those challenges that I can think of are already so complex that I think they're kind of a little bit beyond the introductory lecture. So some of the things might be easier for you to follow than others, and I do apologize if some of the things are over the top, but it's just, it would take me so long to explain the whole background of some of those issues that it would be a little bit difficult to capture it all. But I hope I can give you some general insight. So 3 concrete challenges. One challenge is, well, what's the best information measure to use? So for multivariate decoding, I was telling you that we can use the decoding accuracy. So how well can we predict the examples that we were leaving out? How well can we predict what the actual membership of that class was? But is that really the best measure of information that we can use? This is something that we don't really know. So in multivariate decoding, one alternative measure is that's borrowed from signal detection theory, is using the area under the curve, which would then be a measurement that's less prone to a bias caused by some general shift in the patterns than the decoding accuracy. So let's say you have your separating hyperplane, and here are your 2 different populations that you're measuring, and they're very nicely separated, but now you just have a general shift in the activation. Well, now the classification accuracy could be at chance, but you can still, you know that they're still separable, and with the area under the curve measure, you could actually correct for this shift in the bias, which you can't do with the accuracy. But that's just one possibility, and we don't really know if it really is working better. And maybe continuous valued approaches like assigned decision value are more powerful than accuracy or area under the curve. We also don't really know this. Then recently, alternative encoding-based approaches have been suggested, like the cross-validated Mahalanobis distance. But there's not much that we know about these approaches, and some people have had difficulties replicating the results. So this is like in Walter et al., who was saying that this is a very, very good method to use, but some people have difficulty replicating this. So we don't really know if this is the best approach. We don't know what's the best information measure. In representational similarity analysis, what distance metric should we use? Should we use the Pearson correlation, or should we use the Euclidean distance? We also don't really know which method is better. My personal experience is better with Pearson correlation, but there's no formal comparison between those two. Well, a good model of the noise covariance in the data. So I was talking about the noise covariance earlier, may improve the fit, but we also don't know enough about this. So this is one of the problems. A very important problem that a lot of people are dealing with right now, but that we don't really have a good solution to yet, is that a multivariate decoding and representational similarity analysis are optimized for testing single variables at a time. So you always, when you're running a prediction, you always have one variable that you're predicting. So I'm predicting whether someone is a patient or a control, or I'm predicting. You can also make continuous predictions. I'm trying to predict the age of a participant or something like this. But for decoding, you normally never predict multiple variables at a time. You normally don't predict that. I mean, it's possible there are like multi-label classifiers, but it's quite unusual. And in the functional imaging literature, it's not commonly done. So this is kind of problematic because first of all, we can't look at interactions in factorial designs. We can't look at the unique effects of variables because we can only say, well, maybe the best variable to explain those patterns of responses is the color of the stimuli, or then on top of that I run a test for the orientation, and then I run a test for the shape, and you can't really combine those together in a meaningful way. So you for figuring out like what's the best explanation, how much unique variance is explained by each one of them. And confounds are still very difficult to deal with. There are possible solutions. So for representational similarity analysis, you can use a multiple regression approach, but that has additional assumptions that still have to be tested. For decoding, there is a very recent paper that is still on bioRxiv, where these authors have suggested that you can run confound regression during cross-validation. So that might be a possible solution for confounds. Another possible solution is to completely avoid running multivariate decoding to start off with, and just run like a multivariate GLM, which is an alternative approach suggested by Allefeld and colleagues in NeuroImage. And the third question that I wanted to address is like, what's the correct statistical test to run when you run multivariate pattern analyses? So the current standard, and I can't go into detail why this is the current standard, but at the MVPA level, so when you're asking questions like, does my classifier distinguish between patients and controls significantly better than chance? Or does this brain region carry information about objects in this specific participant? That's like the MVPA level. And there, like the standard approach is running a permutation test or like a randomization test, because like the assumptions of classical tests don't apply. And then there's, on top of that, the second level, which is like normally the group level when you run an analysis at the group. So does in the group of subjects that I have, which you want to generalize to the population, is there an effect of X in this brain region? So does this brain region distinguish between different conditions at the population level? And the factor standard at this level is running like a t-test or an ANOVA or a sign permutation test. The problems are for the MVPA level, for running a permutation test, the assumption of exchangeability is often violated. So this is specifically a case for trial-wise analyses. And the reason why this assumption is violated is because the data that we're dealing with in fMRI has like temporal autocorrelation. And due to the temporal autocorrelation, even within a run, you can't just switch trials as you would normally do in a permutation test. There's a specific structure in the order in which the stimuli have been presented that you can't just exchange easily. And currently there's no statistical test that allows you to deal with this. So when you're running permutation test at the MVPA level, or most of the time the permutation test is the assumptions of the permutation test are going to be violated. At the second level, the random effects test against chance has been shown not to be valid. I forgot to include the reference here, but it's also by Allefeld and Haynes. So possible solutions is, well, people need to develop new permutation tests for dependent data. And I know that there are some people working on this right now. And for the second level, well, you could run a different type of test. Instead of running a classical random effects test, you could run something known as prevalence inference. So instead of looking whether the population of subjects has an effect, you could look at whether you find that the proportion of subjects that are carrying effect is larger than expected by chance. Or you could also run like a random effects test against a baseline condition. So just to sum everything up, I think that's like enough of the open challenges. I just want to give you like a little bit of a timeline of the milestones of MEPA. Like what are the different important studies in MEPA that have been done. So what I think is probably one of the first MEPA studies, and people don't often acknowledge it enough, I think, is a study by Edelman in 1998. Where he was looking at brain activity patterns, and he was looking at how similar the activity patterns were to each other. So this was in some way a precursor to representation similarity analysis, where you can see that, well, for these different humanoid body shapes, you find like similar activation patterns. For these cars, you also find similar activation patterns. In 2001, I think that's the most well-known first multivariate decoding study, where Haxby and colleagues were able to decode with extremely high accuracy what object a person was shown, whether it was a shoe or an office chair. In 2005, though, I think was the moment where there was a breakthrough in multivariate pattern analysis. That was when two studies appeared in Nature Neuroscience, one by Kamitani and Tong, and one by Haynes and Rees, that was showing that you can decode the orientation of gratings shown on the screen with pretty high accuracy. And that was where everyone noticed, oh wow, this is actually a really powerful method, and we all want to use it now. And today, it's one of the mainstream methods. But I think those studies were really where the breakthrough was starting. 2006, Kriegskorte and colleagues developed the searchlight approach, which I mentioned earlier. And then 2008, Kriegskorte also, by the way, this was also done here at NIH, so both of these studies, Kriegskorte formalized the representation similarity analysis approach. Then in 2008, a univariate study was conducted by Kay and Gallon. So this is not a multivariate pattern analysis study, but the reason I'm mentioning this here is because this was the first study where it was able to predict examples that were actually not even included for categories not even included in the test data. So they built like an encoding model, and based on that encoding model, they could then carry out decoding. That's why I'm including this here. And they can decode from the brain activity patterns categories that had not been shown previously. Then in 2011, a study by Haxby and colleagues, also quite influential, where they developed a so-called hyper-alignment approach, which allows aligning the brain activation patterns from one participant to the brain activation patterns from another participant, which now makes it in theory possible to train a classifier on one brain and test it on another brain. And then in 2014, there was a great study by Radek Cichy, who combined fMRI and magnetic fMRI and MEG, magnetoencephalography, by using representational similarity analysis to look at both like the spatial and the temporal information, the evolution of spatial and temporal information at the same time in the brain. So it was quite a groundbreaking study. And then like in 2015, 2016, and that's something that's it's a very recent development, is the use of deep neural networks and relating them also to brain activity patterns using, for example, representation similarity analysis. Okay, so just to sum up, MEPAs are often more sensitive than classical univariate approaches, and it can reveal representational content. It works by combining information across different voxels and also by using their covariation. then investigating brain activity and informational content are two quite different approaches, and I hope I made that, that clear. And we can use cross-validation to reduce bias from overfitting. So if you are interested in reading more about these different topics, I here have some suggested readings. Thank you very much for coming, and I'm open for questions.
So of the different types of classifiers, this is a slide in the sort of way back in the middle. You had linear discriminant, support vector machine, and something else. Which is the least sort of computationally intensive?
Of these here?
Uh-huh.
Okay, so I just want to repeat the question just in case the mic didn't work. So what of these different classifiers is the least computational intense one? Definitely the Gaussian naive Bayes classifier, because there's no need to take the covariance between the different voxels into account. So this will run extremely fast. You can run a searchlight approach in a couple of minutes very, very easily. Whereas this approach would take longer. There's very, very efficient solutions for support vector machines out there. So at least to my knowledge, all solutions I know of support vector machines are more efficient than linear discriminant analysis.